We are privileged today to have as our speaker Professor Sam Black of the Department of Veterinary and Animal Sciences. During his 20 years of conducting research in that department, Dr. Black has made significant contributions towards eradicating animal African trypanosomiasis, a disease that severely constrains both cattle ranching and integrated agriculture in sub-Saharan Africa and serves as a major obstacle to food security in that region. The extent and potential of Dr. Black's contributions have recently been recognized by the Bill and Melinda Gates Foundation through a 3-year grant supporting his research as part of an international effort to develop a vaccine to control and cure animal African trypanosomiasis. Dr. Black is a highly successful and respected scholar. He has published results of his research in more than 100 peer-reviewed articles and in over 10 book chapters. His contributions towards understanding immune response to African trypanosomiasis have been seminal and sustained for more than 30 years. Further, his latest results greatly increase the chances of providing a successful vaccine against a devastating disease, an outcome that would significantly benefit human and economic health on the African continent. This afternoon, Professor Black will present his lecture entitled Approaching the Endgame, Seeking Sustainable Control of African Trypanosomiasis. Please join me in welcoming our distinguished lecturer to the podium.
Thank you. It's a pleasure and an honor to join you today and discuss our work on animal African trypanosomiasis. As you see, this is our parasite, one of several species of African trypanosomes which infect and kill ruminants, cattle, sheep, goats, as well as other livestock animals, particularly pigs, horses. These parasites are protozoa. They're single cells. Despite their rather complex shape and lifestyle, they are a single cell with a single nucleus. And here you see them in their natural habitat, which is in the blood of an infected animal. And these parasites live right out in the plasma of an infected animal in the face of the immune system. They're transmitted by this guy. This is a tsetse fly, Glossina species. There are several species of tsetse that transmit the African trypanosomes. They all have this enormous feeding apparatus, which actually is strong enough to drill through the skin of a crocodile. And they introduce the parasite in their saliva into the skin of their target animal while taking a blood meal, and they flush their proboscis with saliva containing anticoagulants to make it easier to feed. The parasites leave the skin, travel to the lymph, through the lymph to lymph nodes, and from lymph nodes into blood. In the case of some species, leave the blood and travel into tissues, including the central nervous system, where in humans they cause sleeping sickness. The tsetse that transmits the African trypanosomes has a predilection for temperature and humidity conditions that are prevalent in sub-Saharan Africa, in the humid and semi-humid regions. And that is the regions that are most suitable to rain-fed agriculture, and of course, for which rain-fed agriculture is limited because of the limited ability to ranch cattle in that area. The red here is the distribution of cattle in sub-Saharan Africa, the pink, the distribution of tsetse, and green where they both overlap. And you can see immediately that the bulk of cattle ranching is on the fringes of the tsetse habitat. Those cattle which are ranched in the fringe of the habitat where the trypanosome challenge and tsetse challenge is reasonably low, they require chemotherapy or insecticide treatment in order to be sustained, and it costs Africa in the order of $4 billion a year to do that. But that's really not the big issue. The big issue is all that region, Democratic Republic of the Congo and surrounding countries, where you cannot have cattle or other ruminants supporting agriculture. For example, this is the distribution of cattle again, missing the highest areas of tsetse and trypanosome challenge. The same thing for the sheep, the same thing for goats. So when you take that into account, and also horses and pigs, you discover that this central region of Africa isn't going to have Bruegel-like farmyards. It's not going to have little houses on the prairie and wagons carrying civilization forward. It faces a completely different challenge, an ability to grow a society economically under severe constraints of agriculture. In the rural economy of agriculture in probably most developing countries, cattle plays a major role as a source of meat, although it's often too valuable to consume the meat, and more likely people consume the blood and constantly tap that resource. They need milk, not only as a source of protein, but as a source of vitamins, which is critical for cognitive development of young children. The cattle provide traction. They make it possible, instead of going out with your djembe and digging up a little bit of hard clay, to actually get a plow that will make life easier, to grow food easier. They convert inedible vegetation to fertilizer, which can be used to grow edible vegetation, and that is the basis, of course, of integrated agriculture. And they are also a bank. As long as you've got cattle, they're a resource that can be sold, and you don't need some kind of building to put your money away in. And so that is a commodity which is essential to rural development. Again, the Tsetse habitat, centering in its highest density of Tsetse and Trypanosome challenge in the area of the Congo, which you can see is also the highest percent of the population of undernourished individuals. Around 60 to 75% of the population are having insufficient food, and the surrounding areas, while less affected, are nevertheless strongly affected by this poverty and lack of ability to generate enough food to eat. And that is why African trypanosomiasis is arguably the major constraint to food security and food sufficiency in sub-Saharan Africa. In 1979, I joined the International Laboratory for Research in Animal Diseases in Nairobi in Kenya. And this was an institute that was founded by the Consultative Group for International Agricultural Research with the goal of accelerating the development of vaccines that would make it possible to more effectively ranch and produce food in sub-Saharan Africa. And the goal of the institute was to develop a vaccine that induced sterile immunity, that is, utterly prevented infection of cattle with this parasite. Alternatively, we would have been very happy had we made one that limited the severity of disease and still made cattle productive. But there were substantial challenges. The majority challenge of which is the antigenic variation of the surface coat of the parasite, because vaccines depend on identifying a component of the pathogen, an antigen, that is present in all strains and species of that pathogen, that you can invoke an immune response to, a response that will be boosted when the animals are re-experiencing the same pathogen and will occur more rapidly and will eliminate infection. And that can only work as long as you've got a conserved target for the immune system, and does not work very well when the target is as variable as the immune response that would attack it. So here is our parasite in cross-section. This is a transmission electron micrograph of that cross-section. This little pink thing at the top is its flagellum, and that's the flagellum here, the cell body. All the little circles are subfollicular microtubules that give the cell its form. And this fuzzy gray area around the outside of the parasite, that is its surface coat. And you can see it completely surrounds the flagellum and completely covers the cell body. That coat is made of reiterations of a single glycoprotein encoded by a single variant surface glycoprotein gene. In fact, there's approximately 10 million of these molecules on the surface of the parasite, each of them a very short distance apart, about 0.5 nanometers, and held in the outer lipid leaflet of the plasma membrane of the parasite by a fatty acid anchor. Those molecules, those VSG molecules, are highly immunogenic, and they trigger host antibody production. Now, antibodies, for those of you that haven't studied them, are disease-fighting proteins that were made by antigen-specific B lymphocytes. And you have in your body a legion of antigen-specific B lymphocytes, each with a receptor for some aspect of the antigenic universe that constitutes all the pathogens that could attack you. For example, this one here, it might see a little bit of a measles virus. Or this one here, it might see a little bit of a Lyme disease, a bacterium. And this one, the VSG of a particular antigenic type of an African trypanosome. And when it encounters this antigen, it's induced to multiply and eventually differentiate into a factory cell that secretes vast amounts of protein, which is a copy of the receptor initially used to perceive the pathogen component, and allowed to be released free of the parasite because it's genetically remodeled to lack a few things that hold it stuck in the membrane of the cell. And that is called a primary immune response. And in addition to getting that primary immune response, the stimulated cells generate memory cells, which when reboosted are induced to make a second, more rapid, slightly different kind of antibody response, more effective at destroying the pathogen. And that is the basis of vaccination, to get such prime cells so that when they re-encounter their antigen, they'll be better at defending the infected individual. Primary responses mainly inducing IgM antibody, which I'll tell you about in a moment. Secondary responses mainly inducing IgG antibody, which I'll tell you through this talk is critical to the defense of cattle and other ruminants and other mammals against African trypanosomes. This is a scale model of such IgG antibodies and IgM antibodies reacting with the surface coat of an African trypanosome. And you can see that the scale of the antibodies is such that they cannot penetrate between the coat molecules and access the membrane of the cell. Consequently, they can access conserved surface components that are hidden by the coat. All they can see is the very tip of the coat molecules. This is a picture of an African trypanosome stained with a particular antibody, a monoclonal antibody made by all the progeny of a single clone of activated B cells. And that antibody is completely covering the parasite. This is a DNA stain shining through the fluorochrome used to reveal the antibody bound to the cell coat. And that gives you an idea just how the integrity of that coat is, how dense it is on the surface of the cell. IgM antibodies stimulated by the coat are very good at recruiting enzymes from the plasma, the blood plasma, which allows them to drill holes in parasites, causing influx of extracellular fluids and bursting of the cell. And IgM is a lot better at doing that than IgG molecules. IgG antibodies, on the other hand, are really good at putting tags or addresses in the surface of pathogens that cause them to be homed in on by phagocytic cells, specialized cells in our body that eat large particles, that engulf them, add enzymes to them and break down the cells, recycle their usable components, and excrete those parts of the parasite which are not useful and which are difficult to break down. And IgG antibodies are much better at doing that than IgM antibodies. So, highly immunogenic parasites, dense surface coat, really easily killed by antibodies against the coat molecule or induced to be phagocytosed. Why is that a problem? And the problem comes not because this doesn't trigger a good immune response, but because the parasite turns off its expressed VSG gene with a low frequency and stochastically, that is not driven by anything in the environment, and turns on expression of a gene that encodes an utterly antigenically distinct coat from its progenitor cell. So an antibody induced against this parasite cannot react with this one. And the result of that is, if you've got a growing population with a particular kind of coat type dominating, when it invokes an antibody response and that population is destroyed and cleared, by the time that happens, antigenic variants have arisen that seed the next wave of parasites, and the next wave, and the next wave, and so it goes on. And even that wouldn't be much of a problem if that was all that was happening, because simply you would be having an organ that kept changing its surface and was part of your body and engaging the immune system. But every time all those parasites are killed, they release a lot of material that causes really powerful inflammatory responses. These inflammatory responses interfere with the hypothalamus in a way to change your sense of your body temperature, resulting in elevation of body temperature to give a fever. And that will escalate and repeat and escalate and repeat until it disables the individual. Byproducts of the reaction cause destruction of the red blood cells, diminish the oxygen carrying capacity of those cells, and eliminates the cells that cause anemia. They mobilize the body resources, causing wasting disease, also called cachexia. Ultimately, they cause organ failure and typically death of the infected susceptible animal. The repertoire of VSG genes within a trypanosome is very large. Any single individual will have up to 1,000 genes and pseudogenes encoding different surface coats. And additional coat types can be generated by making mosaics of bits of those genes. And so the repertoire of different antigens that these parasites can produce, even from a single infective organ, is absolutely enormous, in the thousands. And if you travel to a different region where you're collecting trypanosomes, you may well find that the parasites there have a different set of VSG genes as their basic 1,000 genes, and the same as you move between species. So you cannot combine these into some kind of assembly of VSGs that will give a vaccine. The diversity is almost as great as that of antibodies that the host can produce to protect itself against them. So it's reasonable to ask, are there conserved trypanosome antigens that can be targeted and better able the host to control all the parasites, irrespective of their VSG coat? So in our lab in Kenya, we dissected what trypanosomes needed to get from their host in order to go through their cell division cycle and grow, that is, multiply. And we found that these large molecules were critical. They absolutely needed the bad lipoprotein and the good lipoprotein, the LDL and the HDL, both of which they considered delicious. And they absolutely need an iron-carrying donor, which brings iron into the parasite and allows it to use it as a cofactor for its enzyme functions. And these molecules are enormous, and this one is approximately the same size of a single coat antigen. And consequently, they cannot find their way through the coat to the parasite surface. The receptors for the parasite to harvest these components from the host have to protrude through the coat and gather them, and therefore seemed logical at the time to be targets for protective immunity. This is where you find those receptors on the parasite. It's called the flagellar pocket. Again, you're looking at a cross-section of the parasite. This is the origin of the flagellum, and there is the flagellum coming out of the cell. And as it beats, it acts like a bellows pump. When it beats one way, it sucks in material from the outside into that pocket. When it beats the other way, it blows material out from the other side. And so this is like a feeding chamber. And the material taken in, perceived by receptors to lipoproteins and transferrin, go into endocytic vesicles that are taken into the cell, broken down and used for the parasite's purpose of growth. So, having identified these molecules that were required for growth of the parasite, we identified the receptors, as did other scientists at the time, and others and us immunized cattle, mice, variety of animals with these receptors, and were utterly disappointed to find that our animals remained as susceptible as before to infection with a parasite, and were no more able to control the depredations of the inflammatory response that the parasite induced, and this is why. When you stay in cells with these antibodies, you find that the receptors are almost entirely internal in the parasite in endosomal structures. This electron micrograph is stained with antibodies conjugated with colloidal golds. The electron-dense gold shows as black dots in the section, and this is where the receptors are, and you don't find them on the surface, and you don't find many of them in the flagellar pocket. And the reason you don't find them there is because in steady-state conditions where they're encountering the food they're bringing into the parasite, they're being mobilized from the surface of the cell into its organelles, leaving so few receptors on the surface that they can't focus the destructive power of the immune system onto the parasite in order to kill it. Secondly, the amount of food of LDL, HDL, and transferrin available in the plasma for these animals is so large that you can't effectively compete the uptake of those molecules by the receptors by anti-receptor antibodies. The net result, targeting these highly conserved, highly essential receptors for the parasite, was a bust. It didn't help any more than trying to understand the diversity of VSGs that go into the parasite. And so at that time, we were faced with the question, is control of African trypanosomiasis just a lost goal, something that we cannot achieve? And in fact, I didn't have the faintest clue what to do next, and decided not to do anything sensible, but just simply look for inspiration from nature. And so I decided I'd look at what mechanisms were available in wildlife selected in Africa by interacting with tsetse and trypanosomes over eons in order to make them capable to carry the disease without symptoms. And the animal model that we chose for this work was the Cape buffalo. And this animal is a migrant bovid. It goes through all the different riverine valleys and plains where tsetse and trypanosomes are prevalent. It's utterly resistant to disease caused by any of the several species of African trypanosomes, but it does get infected, it does carry the parasite, and it does transmit the parasite back to the tsetse fly. Our studies with the African trypanosome were made possible in Cape Buffalo by Jan Groothuis. He's a Dutch veterinarian who worked at the same time as I worked in ILRAD. This is him last year when I visited him, so he's a little bit different from the old days. And he captured Cape buffalo calves in the northern territories of Kenya and brought them down to the vet labs in Nairobi, which at 6,000 feet is outside the range of the tsetse fly and is too cold and hasn't the right humidity to support tsetse and therefore fly-transmitted trypanosomiasis. And so these animals were bred for some generations in this environment, escaping any influence from environmental stimuli that they would have brought with them, and revealing, when you study them, genetically acquired resistance mechanisms. And this is the first experiment we did with the animals. Here's an infected cow, multiple waves of parasites as they change their coat, as we have talked about earlier. Here's our infected Cape buffalo, one big wave similar to that in the cow, and then a cryptic infection that occasionally blips. And this actually is an eland. It's another African bovid that's resistant to trypanosomes. Hugely long, sharp horns, can jump amazingly high. It's a terrible experimental model to work with. And so we stuck with the Cape buffalo. So strengths: Cape buffalo, low parasitemia, no signs of disease. And in fact, after some months, self-cures. Cow, moderate parasitemia, anemia wasting, lots of secondary infections, and often die 2 to 6 months after encountering the parasite. So how do those Cape buffalo control the African trypanosomes? And this is how they do it. To study that, we took advantage of the fact that the rate of antigenic variation of the parasite is relatively low. That is, 1 per many generations of organisms makes the switch. And that number of generations is in the order of 10 to the minus 3 to 10 to the minus 6, so that you would get 1 parasite per 1,000 that has switched its coat. Which means if you maintain parasites by passage in them after cloning, in the absence of an immune response, you can maintain them as 99.9% of one homogeneous variant type. If you put that into an animal that can remit the parasite, you can take its progeny, which have switched their coat, clone them, and by recloning achieve repeatedly many different relatively homogeneous populations, homogeneous with respect to the antigenic coat they express. And so we put 1 parasite into a buffalo, and we find 10 days later there's a ton of antibody against the VSG of that parasite. And if we follow that for months, actually, there's always a lot of antibody present against that trypanosome. And it's almost entirely of this little guy here, the IgG antibody. And sometime later, we look and we find now a lot of antibody against a different trypanosome VSG type, and it's sustained. And later, yet more, and it's sustained. In contrast to the cow. Cow makes just an antibody response that sees each of these parasites also, but it's short-lived. It's not as high a titer, and it's almost entirely this kind of antibody, an IgM. And I mentioned to you earlier, the lifespan of this one in the blood is long. The lifespan of this one in the blood is short. It's well known how that has come to be. I could tell you, but we're not going to go there. We just accept that that's the fact of these antibodies. After some months, if you collect the serum from a buffalo and you purify the infected buffalo and you purify the antibodies from that serum and you add it to tissue culture where these parasites have been trained to grow, we trained them to grow in plastic dishes, those antibodies prevented or killed the growth of these parasites in vitro. If we took different strains of African trypanosomes and different species of African trypanosomes, those antibodies did absolutely nothing to them. Consequently, we were not seeing some kind of conserved element that was present in all the strains. We were seeing clone-specific antibodies, VSG-specific antibodies made by the host. The cow didn't do this. So from that, we conclude that the superior capacity of the infected Cape buffalo to sustainably control African trypanosomes correlates with a superior capacity to make this long-lived trypanocidal, trypanostatic, VSG-specific IgG antibody response. And so the question then comes, why on earth doesn't the cow do the same thing? Well, we didn't study that in the cow for many reasons. We chose to study it in laboratory strains of mice, small rodents that are susceptible to trypanosomiasis, where practically every gene can be taken away or modified so that we understand very much what the genetic basis of host responses are in this strain, and for which there are endless immunological tools to study and dissect the cell types involved in immune responses. And this is what happens when you put our parasite into this particular mouse, a C57 Black 6, and all its identical, genetically identical family. You get an IgM response, which is short-lived, and you get no IgG response, and this mouse will die if left to do that by about 35 days after infection. And after that first response against the first input variant type of parasite, there's no longer a good immune response. So an extreme example of susceptibility, even more so than the cow. If we want to understand how the trypanosome infection modulates that antibody response to make it ineffective, to make it IgM only, and also to actually decline in efficiency with time after infection, you actually have to look not at the antibodies, but at the cells that make them. To look at the cells that make the IgM and the primary response, and importantly, the cells that will make the IgG, that long-lived antibody response in buffalo, is really important for controlling the pathogen. And then you need to know the diversity of populations of B lymphocytes, which are born from a hematopoietic stem cell in the bone marrow, develop in the bone marrow to the point where they have an antigen-specific receptor, come out into the periphery, are selected during transitional phases to become these two lineages. Marginal zone B cells, which respond to hemoparasites very rapidly by making IgM, and follicular B cells that respond to antigen in the presence of T lymphocytes under the right conditions by becoming memory cells and re-stimulated, making lots and lots of IgG. These different populations of B lymphocytes can be distinguished by what are called different sets of differentiation antigens on their cell surface. Now, differentiation antigens are just molecules that do work. And the different molecules that are present reflect functional specialization in the same way that different combat jackets worn by other defense individuals indicate their job in life. You can identify the differentiation antigens on the lymphocytes that we're interested in, the B lymphocytes, by targeting them with antibodies with a covalently associated fluorochrome. You would stick it to the cell, wash away everything that doesn't bind, excite it with a wavelength that excites the fluorochrome, and voila, we have a green cell, in this case detecting IgM. It's one of those early B lymphocytes. But there are multitudes of antigens that dictate what the clonal distinction is between these subpopulations. You need to paint your cells with fluorescent antibodies that see several different differentiation antigens for each subpopulation. And not only paint them with the specific sets of differentiation antigens, but have a way of monitoring how many cells have them and what different antigens are on the cell and what quantity of antigen is on the cell. And that is done using this machine called a flow cytometer. And the flow cytometer blows cells which are stained with antibodies through a nozzle, interrogates them with a laser. The excited light is collected by photomultiplier tubes, picking out the different wavelengths of excited fluorochromes, and they go to a computer that gives you a nice little printout of amount of fluorochrome per individual cell. And each little dot here is an individual cell. So this one has got a lot of this fluorochrome, a little bit less of this one. This one's got nothing of either. And they use color to show when you've got so many dots that you can no longer tell the density of the population to tell you how high the peak of staining is. And that can be translated to a histogram. These cells in the histogram have no staining, intermediate staining, high staining for the fluorochrome measured in this dimension. And this is what happens to B lymphocytes in an infected mouse. And all you need to think about is this population, which is a transitional zone, the transitional cells coming from the bone marrow, maturing in the periphery. The follicular B cells that will be the ones that triggered appropriately will make lots of IgG antibodies. And the marginal zone B cells, those triggered appropriately that will make lots of IgM antibodies. And you can see the transitional cells by 10 days after infection, they are totally depleted, and the same thing's true of the follicular, and the same thing's true of the marginal zone. So the reason you don't make a continued good immune response is because you lose the cells that are necessary to do that. And not just the B cells, but the T cells that provide their help. Here's one population, another population, almost entirely lost by 10 days after infection, substantially depleted by 10 days after infection. What does that mean? Well, we're talking about the adaptive immune system, an immune system that sends out cells to survey its environment, almost in a sense like curiosity, to see what kind of things are happening in that environment, that have got the ability to identify one kind of event in the environment from another through specific receptors, and have memory of that event so that they can respond more rapidly again. In terms of the adaptive immune system, the immune cognitive defect imposed by trypanosomiasis is like losing a very large piece of its brain. So what's left? What's left is an ancient brain, a lizard brain, if you like, innate immunity, the cells and molecules that from invertebrates, from worms upwards, defend against invading pathogens without the discrete specificity and efficiency of the adaptive immune response. It's made up of phagocytic cells that eat and destroy pathogens, and it also releases enzymes and other toxic materials to make the environment really inhospitable to pathogen growth. And this is the sort of stuff they throw out. Hypohalides, hydrogen peroxide, nitric oxide, superoxide anion. They destroy DNA. They eat cells. They migrate into tissues. They dissolve the tissue, turn it into a swamp. This is past Florence Nightingale pointed out that these cells that move in, short-lived and die, all that white stuff, are dead cells at a site of inflammatory response. And they drive up fever. And that's great, because when you get a pathogen in your skin, you want that happening just at that point, because it's going to limit their growth. And those rare cells that have antigen specificity on the adaptive side have time to multiply, reach a critical mass, generate their effector molecules, and get rid of the pathogen, as long as they do that. If they don't, the pathogen keeps doing that until it kills you. And that's what happens in trypanosomiasis. So in susceptible mammals, trypanosomes cause the deletion of the adaptive immune system, massive overamplification of the innate immune system, so we need to know how they cause this disabling immunodeficiency syndrome. And to do that, we use gene knockout therapy, that is surgical removal of specific genes that are required for different differentiation, death pathways and killing pathways in the immune system. And those are created by creating an embryonic stem cell that you modify a gene in, put it into an embryo, put it into a surrogate mother, get an offspring, which is a chimera, cross the chimeras, which are chimeric for their eggs and their sperm, just as they are for other cells in the body, breed true for the ones which lack the modified gene, breed true for the ones that have the modified gene, and now you've got cell lines which are genetically programmed. And you can do that in species in addition to the mouse. For example, here in UMass, cattle were, the first cloned cattle, recombinantly genetically modified cattle were created in 1996 for the first time in the world, just a few months after the famous sheep Dolly was born in Edinburgh. These are all the various gene-deleted mice strains that we looked at over a long period of hard work and substantial frustration, only to find, here we're comparing the ratio of cells in the infected to uninfected control. If the ratio was 1, it would mean all the B cells stayed there. If the ratio is less than 1, you're losing all your B cells, and everywhere you look, the ratio is a lot less than 1, except there. We have this one gene found almost perversely, having done all of these ones, that retained its B lymphocytes. And what does it look like when it's infected? Over 30 days, there's the wild-type mouse losing its transitionals, follicular, and marginal zone, and there's our perforin knockout, keeping them all. In this measure of an antibody response, optical density is positively correlated with the amount of antibody. Here you can see in the wild-type infected mouse, very little antibody, a real pile in the perforin knockout, and the same thing true for IgG antibodies. This assay measures the quality of the antibodies, the simple diversity of antigens within the parasite that the antibodies see. So here, where you've got lots of bands, it means you see lots of antigens, and where you've got no bands, you see no antigens. The wild-type infected mouse, very limited repertoire of antigens of the parasite are recognized by the immune system. Perforin knockout recognizes lots. And that has consequence over and beyond the simple measures of immune function. For example, there's the perforin knockout mouse, and there's no change in the volume of blood comprising the cellular component, the red blood cells, whereas in the wild-type mouse, it's developing severe anemia during the short course of infection. Here's a wild-type mouse infected. It drops acutely its body weight, slowly recovers for a few weeks, and then declines again before its death. The control mice, the perforin knockout mouse actually puts on weight more rapidly than the uninfected controls. The wild-type mouse, a big wave of parasites, a blip, then uncontrolled growth and death by 30 to 35 days after infection. The perforin knockout mouse, less parasites, and then a cryptic infection. A cryptic infection like you see in the Cape buffalo. No anemia, no cachexia, great IgG response, see many things. It looked great. But perforin is a really important molecule in the immune system. Your killer cells use it to destroy virus-infected cells, to kill tumor cells. You can't get rid of it. If you get rid of it, you create a different kind of disease. We don't need to make perforin knockout mice as trypanosome-resistant animals. We need to understand what it is about the parasite that turns on this perforin-dependent killing of its immune system. Perforin is a protein found in granules in killer cells, together with other enzymes which work together with it to kill their target cell. And here we have a target cell, for example, a tumor cell, a killer cell that binds to it, releases its granules, the perforin polymerizes into a pore, creates a tube, and these little enzymes drop in like grenades to turn on the death pathway in the target cell. That is how your cytotoxic cells defend you. There's three kinds of major cell types that make these perforin-dependent killing mechanisms: natural killer T cells, and natural killer cells. In that long desert we walked through, not finding genes that were necessary for deletion of B cells, we discovered that the presence or absence of T cells was completely irrelevant to the B cell elimination. This particular gene knockout prevents the development of T cell antigen-specific receptors, and therefore T cells per se. So two of the major categories of targets of effector cells were not present. Instead, we have our natural killer cell, which binds to its target cell and causes it to burst. Now, just like B cells had a certain repertoire of differentiation antigens that define subpopulations, so do natural killer cells have a differentiation antigen that defines them, natural killer 1.1. And if you stay in a normal population of lymphocytes with natural killer 1.1, you find they're your positively stained population. If you treat the donor of that population with antibody to that differentiation antigen, the cells are killed in their absence. Consequently, if you now infect the mouse from which you've deleted the NK1.1 cells, in the absence of the NK1.1 cells, they keep their B lymphocytes. So it's not just perforin, it's a natural killer cell delivering perforin. When a natural killer cell delivers its granules, molecules that were not previously on its cell surface become accessible on the cell surface. They are inner granule components in the inner granule membrane that, when it fuses with the surface, become accessible to stains with an antibody. And that is a mark of death, a mark of having killed. They are made lymphocytes. There's our natural killer cells from a normal uninfected mouse. None of them have the mark of death on their cell surface. And there's our natural killer cells from the spleen of an infected mouse. All of them have killed, as did all the natural killer cells in the liver and all the natural killer cells in the lymph nodes. So there's a generalizable activation of this normally defensive population to kill and destroy all the other elements of the immune system that are important to survival of the animal. That's a mouse. What about the cow? When a cow is infected with a parasite, you get the repeating waves of parasitemia we mentioned before and the anemia and cachexia we mentioned before. But now we have measured and found that they also lost those B cells capable of making the IgG antibody response. Furthermore, their natural killer cells, although unchanged by a week or so after infection, 14 days, by 28 days are now all degranulating, demonstrating that they've killed in situ. So loss of the B cells, killing by the natural killer cell, somewhat similar to what occurs in the mouse and most likely the same mechanism. Again, we need to know what it is about the parasite that turns this normal effective defense cell against its own immune system. And this is how a natural killer cells defend us. They have a whole array of different receptors, unlike the antigen-specific lymphocytes. All natural killer cells have part or all of that array. And that array receives inhibitory signals or it receives activating signals. And as long as activating signals are balanced by inhibitory signals, they don't kill. But if for a moment inhibitory signals drop it kills. Or activation signals rise, it kills. And during their development in your body, they learn precisely the amount of activating and inhibitory signals in all the cells in your body so that they don't normally kill you. But if a virus comes in, it knocks down some of the inhibitory signals, and now that virus cell gets infected. So for about a year or so, we looked at all the different targets of the inhibitory receptors and looked at all the different targets of the activating receptors on the B lymphocytes, only to disappointingly find that they were essentially unchanged. It wasn't a normal process that was somehow targeting these natural killer cells on its own immune system. And more recently, we came to the conclusion, well, the only thing that's left is it has to be the parasite. Somehow the parasite itself is driving, perhaps directly, the signaling of the natural killer cell to kill the B lymphocytes. And made a lysate of the trypanosomes, an aqueous phase lysate, and find within that preparation, which is largely surface coat molecules, that if you mix it with the spleen cells, all the natural killer cells are stimulated to kill. And in fact, there are very few cells living left in this culture apart from the natural killer cells, whereas that's not the case if you just add buffer or the culture medium to the cells. So now we're winding up. We have demonstrated from the start that trypanosomiasis is extremely complex in terms of the immune response of the host. And we have broken, I think, that complexity down to a single parasite-derived natural killer cell stimulator, which may be the trigger of all the pathogenesis in African trypanosomiasis. The next steps are required to inhibit this particular receptor, perhaps with an antibody, to identify whether it's the same in all trypanosome strains and all trypanosome species, to find whether there's any ruminants that lack a receptor for that molecule or fail to signal from it, because that would be the animal that you'd want to breed up as your trypanosomiasis-resistant animal. Obviously, these are essential to confirm the idea that this single molecule is the key trigger of pathogenesis in the disease. So I want to thank you all, first of all, for having me here at UMass for this period. I'd like to thank our donors, which have kept us going over the years and sustained this project. There's been a legion of helpers since 1979 that have been involved in one way or another in this project, and I'd like to particularly point out Deborah Frenkel here in the second row, who's been with me for 9 years and has made major contributions to our fields of study. And I want to thank you all, really, for making UMass a great place to work and a special place to be. Thank you.
It is my pleasure to present Professor Black with the Chancellor's Medal, which is the highest honor that is bestowed on any faculty member on this campus. And on behalf of all of your colleagues, we're really proud of your accomplishments and congratulations.
Thank you very much. Thank you. And thank you all. Thank you.